Before we start today's interview, could you give a self-introduction on you and also the study you are focusing on to your audiences?
Okay, so a little bit about me and what I do. Okay.
Yeah.
All right. Thank you very much, and nice to be here again. Well, in terms of me, I'm a University of Massachusetts at Amherst, Massachusetts, professor. I'm in a School of Public Health and Health Sciences, and within that, I'm within an environmental health sciences program. So I've been a long time professor here. I've been here 48 years. So I've been at UMass an awful long time, put it that way. But my interest in hormesis, I'd have to say, is even older than my stay at UMass. I actually got my first introduction to the concept of hormesis as an undergraduate back in 1966. I was 20 years old then, and I was taking a plant physiology course. Professor came in one day, and we were doing experiments, but one of the experiments was a dose response study with a synthetic chemical that was an inhibitor on the peppermint plant. And he came in one day, it was November of 1966, and he said that the plants were stimulated. And all the other previous years, they had been inhibited in a dose response fashion. And he didn't know why that happened. And he wanted to know what happened. And did we use the wrong chemical? Did we mislabel something? What was going on here? So he invited the students to come back and try to figure it out after the semester was over. And we had 24 kids in the class, so 24 students. And I was the only one who came back to see him to try to figure it out. And so this started my research. And as it turns out, I was able to show that at low doses, the plants were stimulated, but at higher doses, they were inhibited. And that the reason why they were stimulated in the class was that we had made a mistake when we made up our stock solution with a dilutional error by about 10-fold. And so because of a mistake, I was able to discover that these plants were stimulated at low doses. And he made me, I replicated this study probably almost a dozen times. It was done in soil. Then I did the work in hydroponics so I could avoid the soil and see if there was, I would still get the stimulation, and I got the stimulation. And so I never knew the term hormesis, but we published the paper, and I just called it a low dose stimulation and a high dose inhibition. And so that was in the mid 1960s, late 1960s. And so then I went up and got my PhD and in toxicology. And then probably in 1985, I noticed that there was a conference in California on radiation hormesis. And I read about it. And it talked about a biphasic dose-response relationship. And at low doses, radiation may be protective. At high dose, it was could cause cancers and other things. So I said, well, gee, I said, this biphasic response that I saw so many years ago with peppermint, I bet that biology is biology, and I bet that they're related to each other. So I called the person up who was in charge of the conference, told him of what I found so many years ago, and he invited me to put together a paper on chemical hormesis and invited me to speak at the conference. And that's how I got back involved with hormesis back in the mid-1980s. And then what happened after that was in 1989, there was a debate in the Journal of Science on by two people who were leading the conference. One was Leonard Sagan, the other was this guy, Dr. Wolf, and they called it hormesis, point counterpoint. And when I read their paper in Science, it stimulated me to get reactivated, and I started to organize people to focus. I got a lot of research money, And now it's 35 years later almost. And this is where I am today. So that's how I got started with it, if that's helpful.
Well, that's a quite long journey, and you have been studying on Hormesis for such a long time. So over the decades of studying, what do you think is the general concept of Hormesis? What is it truly is?
Okay, well, the way I define it is that it's a dose-response relationship, and it's a biphasic dose response, but it really, I think, embodies two different things. Most people tend to focus on hormesis as an example of what I'd call adaptive homeostasis. When you stress, if you're stressing a system with a pollutant, with radiation, or with aging, or any kind of biological stress, that the system responds to stress by trying to repair any damage that the stress might cause. And that what happens is that not only does it repair the stress that's in the damage that's induced, but it tends to slightly overcompensate. And by overcompensation, it means that you add a certain sort of benefit to this you make the system stronger. That's what it's really doing. When you stress a system, you can cause injury, but the system responds to that stress and can make it itself a bit stronger. And the hormetic response is really, as I said, an example of I think adaptive homeostasis that essentially what it just makes you stronger when you have a low stress. Now, the other thing that I found is that just about every, I think, enzyme system in cells, that they all show a biphasic dose response relationship. And this occurs for what I call not adaptive responses, but for constitutive responses, those responses that sustain the normal daily operations of life. And their dose response is the same quantitative features as the hormetic response. And so to me, they're all examples of hormesis, this biphasic dose response relationship. And so it addresses both constitutive as well as adaptive responses, and therefore becomes extremely significant in trying to understand how biological systems respond, all the way from bacteria up to humans and everything in between. It's very significant, but it's very difficult to study because the responses are modest, and they're at maximum only 30 to 60%, more or less, greater than background. And this is why people tend to not see it, because it can get lost in background variability. And that's why my initial advisor, when I did that work with peppermint, he made me replicate my study about 12 times in the soil, and then about 6 or 7 times in the hydroponics, because the results were always positive, but they were always 30 to 60%. And he wanted to know, well, was this just by chance, or was this variability, and was it reproducible? And so you really have to have very strong study designs. You have to have lots of doses, and sometimes you actually have to have a dose time response, so you can measure not just the dose response, but how that dose response changes over time. Because if you cause damage initially, you're then looking for a compensatory response and the overcompensation response. So you'll see the stimulation. So it's very interesting, but it's very challenging how you're supposed to do that work. Well, I'd say that and so in the 1990s, what I did was I tried to find most people had at least for hormesis. Hormesis was not very not established at all in the big database called Web of Science that I'm sure you're all familiar with. That very large database in the 1980s only referred to hormesis or hormetic about 10 or 15 times per year per year. So which meant that it was the term really wasn't very well understood. The concept really wasn't very well understood. And so, and at the same time, there was, at least in the environmental area, there was a lot of new developments. The environmental, what I call the environmental revolution, was taking place, certainly in the United States, but many other places in the world. And that was that people were recognizing, how much pollution existed, and they were now trying to actually have correction and improve the environment and have stronger regulations. And so in the United States, there became a lot of confrontation and battles, fights between big industry, what I call polluting industries at the time, and the newly created Environmental Protection Agency that was trying to regulate these. And they debated and they kind of fought over what was the nature of the dose response in the low dose zone, and how could you assess the effects of pollutants on human health and the environment. And it really, they didn't know what the nature of the dose response in the low dose zone was. Was it a threshold response? Was it a linear response? And what happened is that the US EPA in 1975 or 1976 made the decision to accept that they were going to use for risk assessment for carcinogen risk assessment purposes the assumption that the dose response for carcinogens was a linear dose response. And so this was very significant, because for non-carcinogens, they were going to assume that the dose response was a threshold. And so when you have a threshold dose response, then you can say if you're below the threshold, the agent doesn't cause any adverse biological effects. But when you have a linear dose response, then for a cancer, then there's an adverse effect all the way down to as you approach zero. So it was a very controversial decision, I thought, by the US EPA to adopt the linear dose response, and it made the cost of cleaning up carcinogen pollution, contamination in the environment very expensive, extremely expensive. Just 1,000 times, many fold more expensive than trying to clean up the pollution from non-carcinogens. So there are always disputes between industry and the US EPA. They're always fighting with each other over what's the proper regulatory situation. And that's why people got involved with the first Hormesis conference, because it was really put together by the electric power research operations from Japan, as well as the United States. And they were concerned because they were concerned with regulations tied into radiation, but the same kinds of issues were going on in the chemical world. It wasn't just radiation; it was also chemical carcinogens, and that's how I entered into this situation. Was trying to say, well, who's right? Is it a threshold? Is the threshold right, or is it the linear model that's right? And so then I go and look at it and I say, well, actually, I think both of them are wrong. It's not a linear dose response. It's not a threshold response. The actual correct model is a hormetic dose response model. So I said, everybody's wrong and I'm right. And so, but then I didn't have a lot of evidence to prove it. But I remembered when I was young, when I was 20 years old, I did a lot of work on hormesis with these peppermint plants, and I knew that hormesis was real. So it started me on my effort to try to see how general the hormesis concept was, and how reproducible it was, And so over the years, I have had to try to convince the world that the world was wrong and that I was right. And that's been a big challenge when you try to think that everybody thinks that you're crazy and you know you're not crazy, so to speak, that you think that you're correct. And so I would have to say now it's 2024. Many years have passed since this all started, and it's interesting. I mentioned to the class that back in the 1980s, the Web of Science database essentially reported, cited hormesis or hormetic about 10 to 15 times. Last year, it was over 20,000 times. So we've gone from 10 in a year to 20,000 in a year. So the interest and growth in hormesis has been very significant. And it's not just, I got involved with this initially because I was an environmental toxicologist. And so these discussions were within the context of cancer risk assessment, the US EPA and other countries. And now the last 15 years or so, I've been very much focused on hormesis and its public health and biomedical benefits. And a lot of my papers in the last 10 years or so have been looking at how you can improve, and protect your brain, how you can protect your heart and other systems, how you can slow down the process of aging, how you can take. Actually, I'm got very much into I'd say traditional Chinese medicine and looking at the plant extracts and so forth. And actually, I take about 40 different dietary supplements, and most of them are based upon traditional Chinese medicine. So I start my day with and I
it's
do
like a
that.
biohacker.
Yeah, so, and I do that because most of the traditional Chinese medicines, when you actually study them, within the laboratory, they all act in a hormetic fashion, and they act to protect one's health on many different systems. And so I've been and most recently in the last month or so, I published two large papers, one large paper, one concept paper on hormesis and aging in the journal Aging Research Reviews. And but a lot of these that go back really into traditional Asian medicine, and it's interesting. I find that in even though there are a lot of people who are studying hormesis today across all the countries, that the strongest efforts are really coming out of China. And that to me is really very it's exciting to see that. And I've seen it's actually great. And one of my friends, he is from Crete. He has a PhD. He's from Crete. Then he took a postdoc in Japan. Then he has a faculty position in China. Eugenius, you can see on my papers, he's a professor in China. And so it gives me insights into his teaching of and his lectures on hormesis within China and other connections there. So I have indirectly I have some insights into all the research that's going on within your country. And it's exciting to me because I'm really into the hormesis thing, and it's nice to see how actively researched it is within China. It's great.
Yeah, you have just mentioned the traditional medicine, and it reminds me of some fictions. Like in the past, there were some people, some characters that taking toxins and poisons to want to treat themselves and to build a kind of like resistance. Is this kind of behavior, like taking the toxins, also be considered as hormesis? And does the long accumulation of these toxins affect our health?
Well, they can. It depends on the half-life of the toxin and the tissue distribution. But say, for example, I know that some toxic heavy metals, let's say, for example, cadmium. Cadmium, there's a lot of evidence of hormetic dose responses with cadmium. However, cadmium has a very, very long half-life in the human body, decades actually, a very long half-life. And so if you keep taking doses of cadmium over the course of your life, then what will happen is that within a certain period of time, you will pass from the let's say hormetic zone into a toxic zone. And so it becomes what you think was good for you at one point becomes harmful for you in a little bit later point. And so you actually have to understand all of this and how it fits together, and to try to make it work for you. And that would be true for other things. This is kind of crazy, but it's true. But in most countries are really very concerned with lead in the environment, and for all good reasons. But I have lots of studies that show that lead is very hormetic in many different systems. However, like cadmium, if you take too much, if you take it for too long a time period, it stops being hormetic and it becomes very toxic. And this is true, in the United States, but other countries, pregnant women have to be careful with exposure to fish that have methyl mercury to protect the developing fetus. But as it turns out, there are probably a good 20, 25 studies with methyl mercury which show its hormetic dose responses, even in neuronal cells. And so most people would think that, well, that's crazy, that's not possible. But it actually isn't. It's not just possible. It actually is. It does exist. But the issue is it's all. It doesn't mean that just because there's hormesis down there doesn't mean you shouldn't be concerned with potential toxicities. You have to be. But it's just interesting biology to understand how systems respond. Just about every system responds to stress by trying to protect itself. Go back to the concept of adaptive homeostasis. And that is that we wouldn't be here on Earth today if we didn't know how to fight and protect ourselves. I mean, this is really what's going on. And that is that when you're stressed, the body recognizes stress, and it needs to protect itself from threats to itself. And that's where hormesis, I think, in general, fits into the overall spectrum. So people might say, well, why do you believe in a hormetic dose response? Why do you think that the LNT, the linear model, is wrong? And I'd say, well, the reason why I think the LNT model is basically wrong is that biological systems have evolved many ways to protect themselves against all kinds of stressors. And those organisms that didn't develop those systems, they died. They were selected against. The ones that developed them and outcompeted other species, or however it worked for them, they survived. And they ultimately led to, we're the product of all that. And so we actually, when we look at biological systems today, we're very strong, and we're very robust, and these systems, there's all kinds of adaptive systems in every cell type to protect ourselves. It doesn't mean that they're fully protective, but it means that it can handle a wide range of chemical, physical, mechanical stresses that we have. And that's why I think that the hormetic dose response is actually makes the most sense, because it's founded in evolutionary biology, whereas the linear dose response is founded in fear, actually fear of harm, and it doesn't, and it's decoupled from a really better understanding, or using the understandings of what we know about evolutionary biology.
Yes, true. And you have also just mentioned the dose and also the half-life. Like the low dose hormesis will produce beneficial effects, but like, is this effect persistent? Is it related to or like from a longer time scale because of the half-life of different like elements and things? The beneficial effects will decay as it goes.
Well, I think it depends on the circumstances. I think that when you take a look um, at agents that are inherently highly toxic that have a long half-life, then you, as I mentioned, like things especially like cadmium, um, you have to be very careful with your exposures to these. You have to keep those exposures extremely low because you don't want these exposures to build up in the body to become within a toxic zone. So you really try to stay away from those when you and that's why one has to know everything about the agents that you're studying, their tissue distribution, how long they stay in the body, and what their biological half-lives are. You have to know everything so that you can make really the correct sorts of judgment. I'd have to say that the what I mentioned, a lot of these ancient traditional Chinese medicines and traditional other Asian medicines, a lot of these agents that are ingested, they're inherently toxic, and they are designed to protect plants from being eaten, actually. But when we eat them, they can stress our systems. Now they actually do. They a lot of our I mean I try to activate adaptive systems in my cells every day. Every day I purposefully, I have to, I purposefully stress my body through exercise, through the foods that I eat, and I even in my the food my let's say my meals per day. I about X number, I don't know how many years ago, but I started eating only one meal a day. So I just eat a meal in the evening. So I go 23 hours without eating all day long. All day I don't eat anything in between, right? And I do that mostly to stress my systems. So I'm stressing there's some underlying mechanism reasons for this, but everything that I do in life, whether it's I exercise for an hour a day, I do this one meal a day, I take these supplements, and the supplements are designed to provide oxidative stress. They actually provide oxidative stress to upregulate the synthesis of antioxidant enzymes to protect myself against all kinds of oxidative stresses that my systems will have. And I think that this is what I think needs to be done within schools of public health when they're educating their students, and their students have to then go and educate the general public, is to try to educate people as to how they can make their own personal life much more healthy than it's not just that you shouldn't smoke or drink to excess, but there are so many other things that you can do. But all these really come down to, from my point of view, is you have to make sure you stress your body, modestly stress your body to enhance your overall health. And this needs to start at a very young age, because the diseases I mean, I'm 77 years old now, so I'm old, so to speak, right? But and it's when you're old at this stage of the game, for me, many people are developing all kinds of diseases. It's various neurological diseases, the neurodegenerative diseases. They have risks of heart attack and stroke and things, all kinds of these. But you start to protect yourself from these when you're younger. If you want to protect yourself from these and have a healthier lifespan, you have to start introducing hormesis behaviors when you're in your 20s and 30 years of age. Like when you're very young, so you need to be introducing hormesis into your lifestyle now, because when you're my age, now, so many years from now, you'll have a much healthier lifespan and things of that nature.
Yes, that's true. I've just noticed that you have taken only one meal a day. So it's like related to fasting and fasting, intermittent fasting, these kind of things has also been seen as hormesis. And I've tried this as well myself for like a couple of weeks, but I didn't stick to it. But I think it will be better if I just pick it up pick it up again and just try to do the fasting thing, and because it can stimulate our autophagy and it's good for our health, right? And there's another thing that I'm really interested in just now in your talk is just you have mentioned the agricultural purpose, that the ingredients and substances we build for the agricultural purpose to kill the insects, and paraquat is one of them. And it has been proved to extend the lifespan of C. elegans. Do you think it is possible that we use like things like paraquat to extend the lifespan of human beings?
Well, I actually, I tell you the truth, I've seen all those data on paraquat and other agents. They definitely can, in experimental models, they can protect against a lot of different stresses, put it that way. But there are many things that are less toxic, you might less inherently toxic than something like paraquat. I try to extend, I try to prevent the occurrence of, let's say, Parkinson's disease in myself and so forth, but I don't take paraquat. I would take, various types of plant-derived supplement sort of activities that are studied very well, and that actually and you have to watch out for anything that acts hormetically. It has to be in the right dose, because you take too much and it becomes toxic. And one of the nice things about dietary supplements is that a lot of them have a relatively poor bioavailability, and so it's hard to become intoxicated by them. And so when I take my supplements, my wife looks at all these little capsules that I take in the morning, and she thinks that I'm crazy. And then, well, why would anybody do this? And I said, well, that's what I study. But I said it's also these might have a bioavailability of less than 2%, less than 1%, and so it's a question of my biggest concern is making sure that I get enough of the stress to activate my systems, because you need a certain level of stress. But I wouldn't want to take a lead supplement or a caffeine supplement or a paraquat stuff. I'd stay away from all those pollutants, right? But I would find my stressors with other things. For example, the intermittent fasting, the exercise. I do other ways to try to bring stress into my life. Rather than I'd have to say, even it's interesting, ionizing radiation at a certain dose range. This is a very interesting. Before society had things like penicillin and other sorts of drugs to help us avoid problems like pneumonia and things like that could kill you. But back in the United States in the 1930s and 40s, before there were things like antibiotics that you could use in a hospital to treat a sick patient, they really didn't know, they didn't know how to treat people. When my father was born in 1911, okay, so he was born in 1911. My father, and so when he grew up as a child in the United States, if he got sick or something, there were no real drugs to treat him. You got a headache, if you got an infection or something like that, you really, they didn't have drugs to treat people like we have today. You get sick, you go to a physician, they give you a nice drug to help you get over your condition. Back 100 years ago, they really didn't have that. And so Just think if you have, you know, arthritis, or if you have pneumonia, or you have back. If we were born 100 years ago, our lives would not have been nearly as good as they would be today. I mean, seriously, you know. But what happened in the 1930s is people started to use, because they didn't know what else to do. They started treating people who had both arthritis and pneumonia. They started treating them with X-rays and a certain level. And what they found, this is really strange, but if you were in the right dose zone, that in many studies in the United States showed that people who were given a certain treatment of X-rays, they actually survived the pneumonia. And back in those days, half the people died who were admitted to the hospital in the United States for pneumonia, half lived. But if you treated them with X-rays, the mortality went from 50% down to only 8%. It was really amazing. And then ultimately penicillin came and it outcompeted the sulfa drugs and radiation. But it's interesting that I've gone back and looked at this, and if you're right in the right zone, the radiation induces a whole series of it creates an anti-inflammatory phenotype. It induces these antioxidant enzymes like many of the supplements do, actually. And in Germany today, this is Germany, I think, is the only country. But if traditional drugs do not help your arthritis, the German government funds research, funds the payment of treating people who have arthritis with what I call low dose radiation treatment, and it's very effective in reducing the pain and inflammation that's associated with arthritis. And in studies with animal models, if you take 6 or 7 different animal models that have been studied, give them a wide dose range of X-rays, you will find that the X-rays actually decrease the inflammation just as well as a drug does. And it does it in a J-shaped dose response. It's beautiful hormetic dose response. Yeah, and the underlying mechanisms are pretty well understood today. So hormesis is occurring in a lot of different ways, and there's a convergence on how it works, and it typically works by the activation of protective mechanisms, a whole host of those, but ultimately leading to these anti-inflammatory phenotypes that protect us from inflammatory conditions. And the problem with aging is that, you know, and as you get older and older, there's a movement towards much greater inflammatory processes in your body, and that creates the condition for the neurodegenerative diseases, but cancer and other things. And one of my goals, my personal goals, is I want to ratchet down my inflammatory markers. I want to have far less inflammatory markers in my body than otherwise would be the case. And I think that's what I think is a public health message, is that as you're going forward, you know, we have all kinds of these pro-inflammatory markers, and we should try to do what we can do to reduce those. And, you know, you can do that with very little cost. You can do that with your lifestyle and other, and what, you know, some of the things that you eat, but that's why I do what I try to do. It's all hormesis based.
Yes, that's true. And arthritis, there is also something that is less fatal than paraquat, has been proven to treat arthritis, which is the bee venom. Bee venom has also been proved to like treat arthritis, but there's a lot of like sub branches of bee venoms out there. So one bee venom is okay for arthritis. Does it mean that all of the bee venom family can treat like diseases and be helpful to us?
Well, you know, I saw that series of questions that you sent me last week, and I have to tell you that I don't know much about bee venom, and so I'm not going to be. And I didn't. I've been so busy in the last week that I didn't actually go back and try to learn more about it. So I'm going to have to pass on that question. But I'd have to tell you that there are many different things that you can do without using bee venom. And all that bee venom may be great, and I'd have to look at that. But that, for example, I'd have to say that I start my day taking like 1,000 milligrams of let's say fish oil, and I drink, and I actually today how I started my, because I don't have breakfast. I started my day before I came to this talk with you. So at 6 in the morning, I took 1,000 milligrams of fish oil. I took about 750 milligrams of extract from green tea. So it was decaffeinated extract from green tea. Then I took about 1,000 milligrams of curcumin. And I took some ginkgo biloba, and I took what did I take? Ginseng. Yeah, I took my ginseng sample and a couple of other things this morning. And so I start my day with an effort to try to load myself up with something that's going to stimulate the production of move me towards a less inflammatory state, and towards but it does this by stressing your system. These molecules themselves are inherently modestly stressful, but they activate your body to upregulate various types of pathways that will lead to a whole series of antioxidant protection. And so that's how I start my day. And I believe that most people need to do that. And throughout the day I will just continually drink green tea, because I'm going to be, it's the same way of trying to be exposed to something that's potentially beneficial for you. Then at 1 o'clock, I'll go to my recreation center and work out for 1 hour. And so I will, and so I'll go on a bicycle, I'll do something like that. But it's all tied into stressing yourself. And even the work that we do is intellectually stressing, but it's in a positive way. It's intellectually stressing. So it's good for our systems to have what I'll say consistent, modest stress, from a physical to emotional to all sorts of ways. And what we've been brought up in the environmental range is to feel that everything is harmful, even at the lowest possible doses. And even if something causes damage at the lowest possible dose, it doesn't mean that the body doesn't respond to that and protect against it. The body will the body, if there is damage, the body has the capacity to it has sensors. It has all kinds of sensors, and it's measuring change in temperature, changes in structure, changes in oxygen tension, all kinds of sensors, nutrient sensors, every kind of sensor, you can imagine. When you have a modern automobile and you back up, it can sense if there's a person walking behind you. It senses everything. That's what goes on within our cells. Our cells are loaded with all kinds of sensors, and if something goes off within our equilibrium, it creates a process of adaptation, and it helps us to get stronger, to fight that off, and to fight off the next challenge. And I think it's really an optimistic message for humans, if we are smart enough to figure out how to protect ourselves. But humans a lot of times aren't very disciplined, and they don't do their research, and they live very unhealthy lives. It's everywhere. But because living a healthy life, it doesn't necessarily cost a lot of money, but you have to be disciplined, and you have to have knowledge, and then you have to bring those together to actually make that happen. And so it's very interesting.
Yeah, that's true. So apart from those food supplements, do you have other like recommendations or other things that related to hormesis that you think is good for our health, like apart from food supplements?
Well, I'd have to say that the big things, as I was just kind of mentioning, was I think that the best medicine that you can take, the best thing you can do for yourself is exercise. That is, and why I say that is I believe that exercise, the effects of exercise, I think affects just about it affects every organ and every system within your body. So it's a very general stress, which is good. Many of the supplements that I would say, I get the reason why I take so many is that each of them has a different tissue distribution. Some pass the blood-brain barrier, some don't pass the blood-brain barrier, some only go to the liver and the kidneys, others go to other locations. And so I try to understand what the tissue distribution is of every agent that I take, so that I'm trying to make sure that all the systems, the skin, the bladder, the liver, that they're all somehow stressed, and it's just not one particular system that's stressed. So the diet actually is critical, as I mentioned, and the stress within the diet is, I think, important, and that is that the issue of the intermittent fasting, I think, can be very important at a particular age, and to maintain that. I think that I actually think that there are so many there are social factors that are important from what I've read. Just having a positive job is really important. Having a good family life is really important. These things they come together and bring a lot of joy, and also, it's very interesting. I have to tell you this, the project that I'm on right now, and I'll be submitting a paper very soon on it, within days. But through women, as they get into their late 30s and 40s, a lot of women, and certainly in this country, they tend to delay their starting a family until 35 to 40 years old. And as they get older, they tend to their fertility decreases. And sometimes it's harder to get pregnant, things of that nature. And so I've been looking at the oocyte and factors that affect aging. And I have found that area is it's being studied by a lot of people in a lot of countries. And I can tell you for sure that it is enormously impacted by hormetic factors. It's really very interesting. You can protect the oocyte. You certainly can enhance oocyte maturation. You can protect it from all sorts of oxidative stresses and other stresses within a hormetic fashion. And I also have to tell you that, I have hundreds of examples of this, hundreds. And yet none of the researchers, this is interesting, none of the researchers in that area ever mentions the word hormesis. Now, I've done the same thing in the world of stem cells, which is a very big area, wound healing, very big area. And I have found these loaded with examples of hormesis. And in the stem cells, I published 14 papers on stem cells a couple years ago, right? And just about every one of those, and I looked at adipose-derived stem cells, neural stem cells, embryonic stem cells, all kinds of stem cells, right? Almost never do the investigators who are studying these and how to stimulate them, how to direct them, how to protect them, things of that nature. And they show many, many examples of hormesis. Almost never do these people, these researchers, ever mention the term hormesis. They don't have any understanding that this biphasic dose response is a hormetic dose response, and that it's a very general dose response. The scientific community is, for the most part, very, very ignorant of what hormesis is, and I find that it's very frustrating to me because this is my field, and at the same time, it gives me an opportunity to educate these other people. But one of the big challenges is in the world of science, the area of aging and biogerontology has adopted hormesis and the language of hormesis, and so has an exercise science and a few other areas. But most of the biomedical areas, they have no understanding, very little understanding of the hormesis concept. It's very, very interesting. And I think they do that because they're so what I call silo. They're so specialized that they don't read what goes on in other disciplines. And so they have no understanding of anything other than their little tiny thing that they study. And that's all they know. They have no knowledge that this biphasic dose response is a general and it's a hormetic thing, and why it's supposed to be there. The scientific community is really, it needs to be educated on hormesis.
Yes, that's true. It's just like when we went into the medical school, we get a comprehensive knowledge of all the things, but as our study goes on, it has been narrowed down again and again and again, and later on we've been studying just on a tiny part of it. Like you have just mentioned the paper on the oocyte aging, and which is related to hormesis. I'm really looking forward to the publication of that paper as well. And what do you think, Professor, that we can use hormesis to utilize in the drug development? How can hormesis be utilized to optimize the dosage or effectiveness of all the drugs and supplements?
Well, I have to tell you, this is interesting. I haven't talked about this, but for example, in the world of, let's say, anti-anxiety drugs, and that's a very, very gigantic business throughout the world, as anti-anxiety, anxiolytic drugs, anti-seizure drugs, just about most families of drugs. If you go into the clinical, I call it the preclinical literature, with the animal studies, where they're studying these, I'll tell you, when I go into the preclinical literature on like anti-anxiety drugs or anti-seizure drugs and things of that nature, all of those studies, for the most part, they show a biphasic dose response. It's really very interesting. And then when these studies then get tested in a clinical trial, they just take the optimal dose, maybe two, within that protective zone, and then they test it. And let's say that it's successful in humans in the clinical trial, becomes a drug. But though most of the drugs are actually hormetically acting, but the pharmaceutical companies and the research community doesn't even understand that they're dealing with a hormetic dose response. They are fundamentally ignorant. And these very smart people, great researchers, big time companies, they tend to be, they don't understand what we're talking about today. It's very, very interesting.
Yes, there's a lot ignorant on the hormesis and the overall concept of it. But like from the very beginning, like hormesis, when we think of this word, we are related to like toxins and poisons, right? So from the very beginning, how human beings get noticed and come up with the idea of treating diseases or like the lifespan issues with these hormesis, is it out of coincidence?
Well, it's human behavior is really hard to understand. It's experimentally, you know, I mean, I've gone back on Hormesis to try to understand its history, and I'm not sure if you've had a chance to read some of the historical papers that I've written, because I could send those to you if you'd like, because it really the problem of, you know, Hormesis had a it was really first conceived of, you might say, experimentally shown back in the 1880s by this fellow Hugo Schultz, and with yeast. But Hugo Schultz, he did really good work, but Hugo related his work to the medical practice of homeopathy, and homeopathy was very controversial, and it was very competitive in the late 1800s with what I call traditional medicine, and the concept of hormesis was rejected by traditional medicine because Hugo Schultz had associated it with homeopathy, and so for most of the 20th century, the medical community was very biased towards the concept of a biphasic dose response, and plus the biphasic dose response is very hard to study because you need lots of doses. You're only going to see a modest response, 30 to 60%. You need lots of samples, lots of subjects that you're going to need to be studying, and it's harder to prove. And so for a lot of reasons, hormesis was difficult to successfully make it into the biomedical literature. It's doing it now because of all of our work, but nonetheless, you know, there's an awful lot more work that the next generation has to do. Your generation has to do to move this thing forward, really. But you've got, you know, you've got a lot of, you know, you're on the right track because there's a lot of evidence now to support hormesis. You have mechanisms, you have reproducible findings, you have good applications. You're in a good position to do some great work, actually.
Yes, that's true. Like, Homie, you have just now, like a few questions earlier, you have mentioned radiation, use radiation to treat arthritis, but there is also some problems right now these days. Like when we are doing the radiological examinations, and so we might get into about those nuclear power plants. They might there's some accident happening and some contaminated water being released. So what are these unexpected, like high dose of these toxins relate to us, and how will it affect our life?
Well, in general, I think you have to look at everything pretty carefully. I tend to think that a lot of what happens in the environmental domain is everyone, as I said, prefacing it by you really have to look very objectively at what's actually happening. But a lot of times these situations become very politicized, and so you have different groups that look at things not scientifically, but they look at them, I call it ideologically or politically, and that to me is the wrong way to do things. But that's what actually gets reported certainly in the United States media, which is very politically oriented. But they don't actually do a good job on getting down to what is the scientific basis for either concern or lack of concern. So each one of these issues needs to be really looked at very carefully, very objectively, in a very non-biased way, so that the public can actually be educated properly rather than be pulled to one side or the other, mostly by politically motivated groups. And so I'm very opposed to politically motivated groups when it comes to science. Science needs to be, you know, follow the science, and then it'll lead to much better answers, rather than you see a lot of people, they believe they know the answer, and then they try to fit the science into what their beliefs are. and it needs to be the other way around. Your beliefs should come from your scientific data, and then you can make good decisions. When you think you know the right answer, that's very bad start. And so I think that when you educate students, like you're in an educational system, it's really important for them to create hypotheses, test hypotheses, follow those hypotheses, and then don't be afraid to be wrong. When in my whole life as a professor, I had maybe 100 graduate students for getting their doctoral dissertations and other things, master's theses, and I would have to say that about 60% of those dissertations proved my ideas were wrong. Okay, when they actually, I thought I knew something, but when they tested these ideas in the laboratory, the students showed that my ideas were wrong at least 60% of the time. And so, and I thought that I was very knowledgeable. But yet, and so I think that society needs to be very careful, and with information, always has to be very objective about information. And this way we don't get, we make less mistakes, we're more objective about it, and we're less emotional when it comes to environmental and other sorts of facts. Try to get the facts right. And even on, the kinds of things that I'm talking about, the fasting or this supplement, I do not take a supplement until I have researched it enormously. And so everything that I take, I never look at the advertisements that a company sells. I get all the scientific papers, and I evaluate the scientific papers. Then I make a decision. Most people can't do that because they don't have our training. See, we're trained to evaluate things, right? But other people, most people can't do that. I mean, my wife couldn't do it. She's like a fifth grade teacher. She teaches English. She couldn't do the science that I do. She has to rely upon somebody else's judgment to tell her, or other people do. So we're very lucky that we are trained in the sciences, because we can actually evaluate things. Most people can't do that. Most people, they're totally, they might be smart and they may know something about other things, but they can't evaluate what you and I can evaluate. It makes it really, it's an interesting situation to be in.
Yes, that's true. Science is about like facts, but our our interpretations might be subjective, and when we impose those subjective interpretations to science, it's actually not a very ideal action. So we need to choose it and take it objectively, and just to look at the facts and then decide what we will go on and doing things. It's a very good way to educate us and to tell us what is currently right and what is currently wrong. That's what I think.
And it's a good thing not to be afraid to be wrong. Everybody makes mistakes, but it's good to be objective, and that's the goal, and to make it less emotional. Stick to the facts. I think that would be my biggest message, really, and that's true for Hormesis. Be as critical on Hormesis as I'd be on anything else. Just always stick to the facts, and see if it convinces you, or how to test something to see if it's correct. That's very, very important.
Yes, and you have been always like not challenging, but like trying to find different things in the scientific world. Like one of your research background has been focusing on cancer, and you have also highlighted the flaws or the insufficient of the LNT model that has existed in the cancer's assessment method. So what, in your opinion, Professor, that can be a very comprehensive and accurate cancer risk assessment that we can carry out right now?
Well, I'd be happy someday to give a talk on the LNT thing to your group if you're ever interested. But I would have to tell you that my evaluations of LNT came out of my work on Hormesis, because I was studying dose responses, and so I ended up, needing to look at the LNT history as well. And what I found, just very briefly, was that the LNT model that really was came out of work in the United States during the 1940s and 50s and 60s is actually fundamentally flawed. It's loaded with mistakes. It's loaded with terrible bias, and it's also loaded with the deliberate misrepresentations of the scientific record. This was carried out by the U.S. National Academy of Sciences. The journal Science participated in these sorts of things, and what's happened, and then to make it all worse, the U.S. regulatory community, like the EPA, adopts these the mistakes and perspectives, and they never go back to actually researching the historical foundations of it. They adopted the biased recommendations of previous U.S. National Academy panels, and I think that they got the dose response wrong because of their biases. And so what's happened is that here I am now, it's 2024, and I'm saying that the entire U.S. regulatory program is totally flawed and corrupt, and so they don't like me very much, although many people do. And but the thing of it is I publish a lot of papers on this. I published probably 100 papers on this history of LNT, and I believe that my story is a correct story, and I'd be happy to if your class is interested, give a talk on this sometime in the future. It would take about 2 hours to tell you the whole story, but I'd be happy to do that if people are interested in hearing my view on the historical foundations of cancer risk assessment. It has lots of implications for every country in the world, including China, and I believe that every country's wrong. Every country is adopting LNT, and I believe every country is wrong. I think they should listen to Calabrese on this matter, and then see where we go from there.
That would be great. Maybe in the future there will be this kind of chances that you can elaborate on the LNT model as well. And today you and I have been discussing for like more than an hour. So there is also many audiences that are willing to ask you some questions and their opinions on hormesis as well. Can I hand these questions to you as well?
Sure, why not? We're here. Let's do it.
I've received one message from our online audiences. You can just see that they have been typing in the chat boxes. I will translate it for you. So the first question is, free radicals are always put on the hat of evil, but a lot of research show that free radicals also has a beneficial side. What do you think of free radicals as poisons? Do we need to take antioxidants, or what antioxidant do you recommend if necessary?
That's actually a great question, and it's fundamental to the whole discussion today. And that is that I'll give you an example that'll answer the question. There was a study that was done with respect to exercise in humans. It was a human study, and people were studying humans that had type 2 diabetes, and so they wanted to have them exercise because they felt that exercise could improve the type 2 diabetes condition. And so they also gave them a high dose of vitamin E. Okay, so they had a group that got the control group, and a group that got the different sorts of conditions. And what they found was that the exercise was very protective. But what they found was that when you gave the people who exercised the vitamin E, which is a strong antioxidant, when you gave them this along with the exercise, it totally blocked the protection and eliminated the protection that the exercise gave. So you think that you were doing something good for you when you exercised and you took the vitamin E, but the vitamin E totally blocked the protection of the exercise. And it relates to the person's question of free radicals, because the exercise is actually inducing is actually creating reactive oxygen species, or free radicals, that are activating some of these pathways that induce the protective effects. And the vitamin E comes in, and the vitamin E can actually block that. And I'd have to tell you, I stopped. I was a reviewer of that paper. That paper was originally submitted to the New England Journal of Medicine, and after about 6 months was rejected. And then it was submitted to the U.S. The Proceedings of the National Academy of Sciences, where it was accepted. It's now been cited about 1,000 times. It's a very big paper. But as a result of that, what I did was I no longer take vitamin E supplements, because I exercise all the time, and I want to get the benefits from exercise and these other factors that I have, and I don't want something coming in to block that. And so I want to have a low level of free radicals operating. I want to have this low level of generated ROS, or reactive oxygen species, within my cells. I don't want too much, but I want enough that activates processes that are necessary to then give me a very strong, protective antioxidant response. I think we want that response to be enzymatic as compared to through chemical.
Yes. And thank you for your answer. I think that the audience will be very glad for all these messages that you have conveyed. And right now I see that one of our audience have raised his hand. So let's welcome Yizhuo. You can unmute yourself, and this will be the last question today. You can unmute yourself and just talk to our professor.
Thank you very much for making me the last person to talk here. Professor Calabrese, thank you very much for the fascinating talk. I was very intrigued that you mentioned you only eat once a day, and you're taking 40 substances. Right. So my two related question is, so with those 40, the combination of the 40 substances, what is the methodology? How do you choose those 40 substances? And if it's okay for you to share your recipe with us, and would you recommend the same for us to take if we're able to do the fasting? That's my question. Thank you.
Well, that's a big question, but I'll try to answer it. Okay. What I've tried to do is, I'd have to say each one of these, I have actually done a major literature review on each of them. Probably at least half of them I've actually now published papers on. So most substances that I take, I've published a paper on. I've done a lot of work on it, and have evaluated it from a hormetic perspective, and then go forward and publish on it. So I actually do that. But what actually drives me here is that I'm very interested in their bioavailability. I'm very interested in their tissue distribution, and many other factors. And so it's kind of putting together supplements that are very complementary to each other. Some are able to reach the brain, some can't. Some distribute are only located within the GI tract and the liver. Some go to the lung and so forth. So I have this big matrix thing on my wall at home, and it's telling me, the relative capacity for agents to be distributed throughout, let's say a mammal's body, and that actually it helps me decide the matrix of supplements that I take. I try to take them so that they complement each other and present a unified whole. So that's what and also I try to do it so that and I'd have to tell you, every 3 months or so, I have my blood tested to see if I've caused any toxicity to myself. So I'll have liver enzymes and other kinds of things tested, because I'm trying to make sure that I haven't taken too much, and I don't want to go too high on that dose response spectrum, so that I leave the optimal zone and go into a toxicity zone. And so so every 3 or 4 months, I'm having a blood sample to make sure that I'm not toxic. This is like how a scientist think. The general public wouldn't do that, that sort of thing, right? They just want to know, will this work or won't it work, and things like that. But and so this is how I go about what I do. I'll be very happy to send my recipe of supplements, and you can check those out, and you can write back, and we can have a dialogue. And so you can say, well, I like what you're doing, but I don't like this. Why don't you try that? And so we could have a discussion about that at the personal level.
That'd be great.
And for anyone who's listening, if you have questions to me, or if you want anything that I have, feel free to email me. I'd be happy to hear from you, and happy to send you different things. So, and happy to meet you sometime. And if I'm lucky enough to make it over to your great country.